All right, hello and welcome to Bioconductor 2021. This is the package demo session for the chromatin segmentation analysis in R. We'll start the demo shortly, but first a few housekeeping items. If you have questions for the speaker, please enter them into the Q&A tab. You also have the ability to upvote questions for the speaker to answer. And if you would like to ask your questions live, please use the raise hand feature, and a moderator will bring you onto the stage so you can ask. There are a variety of emoticons available to give feedback to the speaker as well. And if for some reason you have to leave early, a video of the session will be available a couple of hours after the session has completed. We're gonna have questions at the end, but feel free to post your questions into the Q&A tab all throughout, and we'll just answer them at the end. If you have more questions than what we're able to answer today, please feel free to message Mahmoud, just using like the messages feature inside AirMeet, and potentially he can answer your question there, or potentially you guys can set up a time to meet outside. Without further ado, it's my pleasure to introduce Mahmoud Ahmed from Gyeongsang National University, and he'll be demoing the segmentr package. All right, take it away, Mahmoud.
Thanks, Glenn. Thanks everyone for being here. So I will start by sharing my screen. Okay, is the screen, can you see the screen share now?
Yes, we can see it.
Okay, thank you. So this is going to be a demonstration of a package called segmentr, which is to perform chromatin segmentation analysis in R. So my name is Mahmoud Ahmed, and I'm a postdoc at Gyeongsang National University in South Korea. So this code grew out of a project where I needed to do chromatin segmentation on a set of histone markers for my research. And then I figured that I need to integrate the tool that I'm using to do the chromatin segmentation into my analysis workflow, which is mainly in R. And then I figured that I could write this wrapper and then submit it as a Bioconductor package to get a feedback on it first, and if it's useful to other people, they could be able to use it. So very simply, chromatin segmentation analysis is that you take the chromatin or the genome and divide it into regions, and then look at which histone markers are present or absent in each of these segments. And then you can use that to interpret it, depending on what these histone markers mean biologically. So I divided this presentation into 3 parts. In the first part, I will be introducing very briefly the segmentation analysis and a tool called ChromHMM, which is probably the most widely used tool to perform chromatin segmentation. And then I will apply this analysis to a test dataset and explore the output using the functions in the package. Finally, I will go through the output as it exists in the segmentr package, and then go through different functions to manipulate the output and visualize it, and do some basic summaries on it. So the underlying model that ChromHMM uses is called hidden Markov models. And very briefly, a Markov process is a process with unobservable or hidden state that can be modeled with dependent observable process or data, like histone modification or DNA binding protein data from ChIP-seq data. So the chromatin configurations, for example, an active transcription or a strong transcription on the gene region or a promoter area, are the hidden states in this model, and then they are modeled by the different markers that marks the histone, and that we know biologically it could mean that there is an active transcription going on here, or that there is the lack of markers in a given region would mean that this area is not actively being transcribed or used. So this figure illustrates this process very simply. So the hidden states that I'm talking about here would be the x's, and then the observations that we have would be the y's. So in this case, these are the states that we're trying to figure out, and these are the data or the histone markers that we could use. And between those, there is we could calculate probabilistically how the presence or the absence of these observations mark different states, and how these states could transition between each other. So again, the ChromHMM is a tool that is written in Java, and it's very widely used to learn chromatin models or chromatin states from ChIP-seq data, especially specifically of histone markers. So again, the states are modeled at the combination markers on different regions of the genome. And then a multivariate hidden Markov model, or HMM, is used to model the presence or absence of these markers. And then the combination of histone markers at a given location is interpreted to a biological state depending on what we know about these markers. And this paper here introduces ChromHMM and the idea of chromatin segmentation and apply it to a real data set, which I encourage you to go on and check. So the goal of the segmentor package is to call ChromHMM from R using R syntax, and then to capture the output into R objects, Bioconductor objects that you're familiar with, and then to interact with the model output for the purposes of summarizing or visualizing this output. So this package is available now in the developed version of the Bioconductor, so you can install it using the BioC manager. If you're using the development version of the Bioconductor, and later on with the release, or you can install it using the remotes package from Bioconductor directly, from GitHub directly. So to give you an idea of how this code look like, there is a wrapper function called LearnModel. Does pretty much most of the work that this package do, and you call it with the you have an arguments that you could you would use to pass all the inputs and all the that you need to perform the chromatin segmentation, and then this will call ChromHMM, read the output, and then return it into an S4 object that we will explore later on. I will be through the details of how to call the details and so first, the inputs. The inputs that are required by ChromHMM are two types. The first type is the binarized signal files from the ChIP-seq data, and I will be talking about how to generate those in a minute. And then the second type of input, of required input, is the different genomic annotation file. So the first one and the most important here is the chromosome sizes. So this is just a TSV file with the name of the chromosome and the lengths of the chromosome. And this is important for more than one reason, mainly to be able to segment the chromosome lengths into equally sized bins. And then there are other files that are required. One of them we call the coordinates, and these are the genomic features that you would be interested in performing enrichment or overrepresentation analysis on it. Say to be able to say a particular state is overrepresented in this genomic region, and so on. And the anchors are a similar idea, but instead of using a feature with a start and end, this is just a single location on the genome that you'll be interested in to perform enrichment or overrepresentation around this particular location. So I will show how to generate, or I'll show examples of the annotation files here. So I'm using the human genome 38, and the coordinates, as you see here, could be CPG islands, could be the coding sequences of the genes, could be the transcription start sites, could be the exons, and so on. So these are the types of coordinates that I'm talking about. And it's just a bit file, and with the chromosome name, the start and the end site of this particular region to perform the enrichment analysis. And then the coordinates, the anchors are a similar idea. So here I'm using the transcription end sites and transcription start sites, and these are just like a single location on the genome, so we can perform enrichment around. So this is the chromosome size, and the most important size file. This the tab delimited file. First column, you see the chromosome name, and here the link, and the chromosome. Files that you generate from ChIP-seq, these are just text files, and then usually we generate one for each chromosome to make the size manageable. So the data from the ChIP-seq is counted, but instead of counting at one base resolution, we count in bins of a
Hey, Mahmoud. It seems like that we're having trouble with your connection. I don't know if you're on the low definition already on your camera, but we might want to try that to see if that helps stabilize. Looks like we lost Mahmoud, but he should hopefully be back soon. Oh, great. Looks like he's reconnecting. All right, Mahmoud. Yeah, looks like you're back. You may want to turn off your camera just so that we have your audio, and maybe that will help your connection.
Okay. Yeah, I also lowered the resolution.
Okay. Yeah, if you can go ahead and share your screen, let's see if we can do that. That looks good. Yeah, we got some thumbs up, so that's good. All right, and I think we might have been on a previous slide.
Okay.
I think that's right. Is this okay to start with, everyone? Just give another thumbs up if this is good. Okay, great. All right, yeah, let's go ahead when you're ready.
Okay. Sorry everyone for that network issues. So I think we left from here. I got disconnected in this slide. So again, very quickly, the second input for ChromHMM is the binarized sig files, which you generate from ChIP-seq, and there are two functions to generate those. So the idea here is to divide the chromosome into different bins, and then count the reads on each bin. And if it's above one given threshold, you give it 1. If it's below it, you give it 0. So the example here would be for using binarized BAM is the first the important input here is that you need to make a file, a tab delimited file, to tell ChromHMM which what the information about the file is generated from this particular cell line, and using this an antibody, a ChIP antibody for this histone marker, and so on for all of them. And then you call binarized BAM, give it to the first input is the directory with the BAM files, and then you give it the chromosome size file that we just created, and then the chromosome size file that we talked about in the previous slide, and then the cell marks file that we just generated, and then give it an output directory. And then the output would look something like this. So for each bin, there is a 0 or 1 value for each of the chromosome marks. The model learning is implemented in a wrapper called LearnModel, and this wraps the Java module that learns the chromatin segmentation model from of different states that you choose. So the LearnModel function takes as an input the bins that we just generated in the previous slide, and then an output directory, and then the 3 genomic annotations that we talked about also, and then a number of states that you have to choose, and then some other information on how on the experiment itself, or like the samples that are used in the study. So these are the cell lines that were used in the study, the assembly of the genome that is being used, and the type of annotation, and then the bin size in base pairs. And then the output is, so this function calls ChromHMM, and then it reads the output in an object called segmentation. It's an S4 object that has different slots, analysis slots, in the next slide. So just like in other Bioconductor objects, there are accessors to access the information within these objects. So for example, if you want to access the emission probabilities, you call the function emission on the object, and then it returns the emission probability matrix. And for some functions, because if there is an object or if there is information that is related to a particular piece or a cell line or a sample, in this case, you can just call an argument, call the accessor on the object, and then say I want the piece related to this particular cell line, not everything. I also wrote some methods to interact with this object. So for example, there is a show method that prints a brief summary of the object, what is inside it, and then some instructions on how to access the different slots in the object. So here we just our different slots contained in the object map nicely into the type of information that you can get from ChromHMM anyway. So I will explore it by going through the different accessors with the different slots. slots. So the first one is called emission, and the emission probabilities are the frequencies of a particular function uses the heat map function from the complex heat map, and this can be customized in many different ways depending on what you want to visualize. So this is of the previous code. Which is the enrichment or overrepresentation of states between in a particular feature that we passed in the coordinates arguments in the LearnModel function. So the overlap slots contain the fold enrichment of each state in the genomic coordinate, the number of bases in this annotation, and then divide that by the number of bases in the annotation and by the number of states in the number of bases in the gene is enriched in a particular location. So here I'm using the plot heat map directly to show the overlap enrichment, and then I'm giving labels, like easier labels to read for the genomic coordinates that we provided before. And this is by default calculated for every cell line or every condition that you show. So these are the states in the rows, and these are the genomic coordinates. And then every cell tells you what's the overrepresentation or the enrichment of this state in this particular feature. So the most important output that you can get from chromatin segmentation is the segments. So these segments objects contains the state assignment. So for each bin, which state is it assigned to? And for each cell or a condition, you get a GRanges object with the chromosome name, start, and insights, just like a iRanges object. And then because it's a GRanges object, you can add metadata. The one metadata column that we add here is the state. So you can get this information.
Mahmoud, it looks like we lost your screen share again.
Sequence name or the code. So it went to the promoter of this gene. So
Mahmoud, it looks like we lost you a little bit there.
I'm using the
I think we lost you on slide 26, where you were talking about the most important thing
known
from
gene
a
information,
segmentor was the
ID38, which
segments.
is the AC81. I was using this to stream and think of it downstream. Am I disconnected again?
Yeah, I think you did. We lost you, I think, on slide 26, where you were talking about the most important thing that a segmenting package can get you is the segments. And then you were discussing those, talking about the ranges and things.
Okay. Okay, so sorry, network issues again. So I guess we stopped here. So the segments object or the segment slot from the object contains the state assignment, and it comes in the form of a list of list of G-ranges objects, each one for each cell or condition. And then if you call the accessor on the object, you get the full list. Or if you choose, you can choose a particular cell line to show their segmentation. And this is how the output looks like. So you get the G-ranges object with the chromosome name, the start and the end sites, and then the states assignment. So to visualize this information, I'm using the different Bioconductor annotation and visualization tools. So loading the organism package for the human genome 38, and then I'm extracting the gene information or coordinates from this object, filtering by the entry ID for this gene. And then I'm resizing coordinates to a promoter region of 10,000 kilobase pair around the transcription start site. So I'm using the AnnotationTrack function from gviz to render the ranges and track the as tracks and group it by the state column in the granges object. So loading the gviz and iranges, and I'm looping over the different cells, the different granges objects, and then give the name to make it obvious in the visualization. So the function cells will give you the names of the items in this list. And then I'm subsetting by subsetting this objects or the segments by just the promoter region that we just selected, and then calling AnnotationTrack on the output of the subset, and then group by the states column. And this is how the output looks like. So the PlotTracks function takes a list of items. In this case, I'm passing an item for one cell line, and then it shows you in the squares this full region was assigned to this particular state. So other tracks can be added to make these graphs more informative. So here I'm using the ideogram tracks and the ideogram track to show a graphic representation of chromosome 11, and the genome axis track to show the different the locations that are being shown in the graph, and then the gene region track to show a genome model of the transcripts, introns, and exons of the gene. So to generate the ideogram, you can just call ideogram from gviz on the human genome 38, and then say chromosome 11. This gives you the graphic representation of the genome of the chromosome. And then the G track just adds the genome axis to the plot. And then GR track is you can use the biomarker gene region track from gviz, and this will extract the information, the coordinates of the exons and the introns and the transcripts of the gene that you request. And then you can put all this together and then you tell the function where you want to start and where to end, and how to group the annotation. And this is how the output looks like. So this is the ideogram, this is the genome axis, this is the gene region track, and these are the annotation tracks that we generated earlier. So that gives you an idea of how the different regions around the transcription start site of this gene are assigned different segments. And then I'm plotting the annotation track for 2 of the 5 cell lines, and then you can compare easily between them, see what are the differences and the commonalities. And then if you like, you can perform some summaries, some simple summary on these segmentation objects. So one example of this is called GetFrequency, and this simply counts the occurrence of states across the genome, and then returns an output in tabular or graphic format. So if you just call GetFrequency on the segment object, and then you get a data frame like that. But if you call PlotTrue on the same function call, and you say PlotTrue and pass other graphic options, then what you get is a bar plot with the different segments. So this on the x-axis you see the cell lines, on the y-axis you see the state fraction, or the number of states in each cell line. And then there is an option also called normalized. If you give it true, if you make it true, then it will normalize by the total number of states. So this is a bit easier to read, or especially to compare between different cell lines. So one important thing that you probably need to do if you perform chromatin segmentation is to be able to compare multiple models. And at least in my case, the main issue was having to choose a number of states. So in this case, I can learn the model with different number of states and then compare them, see which one fits the data better. So in this case, I'm showing an example of running LearnModel using lapply on different, and then I'm passing the same arguments to LearnModel, just changing the number of states each time. So the return here will be a list object of each item is a segmentation object. And then compare models takes this list of segmentation objects and returns a vector of the same length with the different summaries. So the default is to compare the correlation between the emission parameters in the states. And only the highest state correlation with one of the states in the biggest of segmentation objects return a vector relate. And then in this case, in the model, and the same way related to there is another in the bigger model. And then there is another likelihood that ChromHMM calculates. So you can also compare models on the object on the list, and then the function will extract the likelihood from the model and return it. I plot this in a point graph. So here I'm plotting the correlation, the state correlation, and then the model likelihood together. So the different states, and then the model likelihood in this case on the y-axis. So you can see how the different models perform compared to each other. So finally, I want to make some final notes. So the first is that the emission and the transition probabilities that shows the are here to show the frequency with which each histone marker or a combination occur across the genome. But the meaning of these states depend on the biological significance of the marker associated with particular configuration, like the index. So some markers with something being actively transcribed or being repressed. The fold enrichment that we calculated in the overlap enrichment or the enrichment around transcription, the transcription start and end sites can be used to define the regions in which some states occur more than other compared to other regions, or how they change in frequency between different cells and conditions. So to either visualize the different kind of biological states that occur at different regions, or you could investigate particular genome, particular gene, or a particular region. You all for your attention, and say sorry again about being disconnected a couple of times. And you can visit this GetFrequency and leave me a comment. So thank you.
That was great. Thank you, Mahmoud. Appreciate your talk. If anyone has any questions, feel free to ask them. Otherwise, what I might do is just ask Mahmoud to maybe go into a little bit more detail about the fold enrichment. That seems super interesting. I just want to learn.
Yeah. Okay. So the idea here is that, well, I call it enrichment here using the nomenclature from the ChromHMM, but it's really an overrepresentation, right? So they calculate the number of bases in a particular state and divide it by the number of bases in the annotation. So for example, if I'm calculating on the CBG islands in CBG islands in general, so I calculate the number of bases in each of these states, and then divide it by the number of bases in the CBG in the feature, the CBG island, and then divide that by the number of bases in the annotation by the total genome. So just like a simple overrepresentation.
I see. Okay. Yeah, that reminds me of more of what I've done with like DE-seq and stuff like that.
Yeah.
Yeah. Interesting. With the hidden Markov models, it makes me think of some other things like MCMCs and things like that. What made you settle on the hidden Markov models? Is that just because that's kind of the standard in this space, or were there other reasons?
Yeah, so there are a couple of tools that they use the hidden Markov model. One of them is ChromHMM, the other is called ChIP-seq. And okay, so to be perfectly honest, I also tried other tools, but they take forever to run, right? So
Yeah.
for example, Segway, it uses like Bayesian networks, like dynamic Bayesian networks, and it takes weeks to run on the same set of data. So this seemed like I don't know, the standard that people are using, and it's more practical.
Yeah, that makes sense. No one wants to wait weeks.
Yeah, I mean, I did wait weeks, and then just never again.
Yeah,
I ran it
do
on
it.
in a data set, but yeah, you need to be able to iterate, right?
Right. Okay, that's great. Does anyone else have any other questions? All right. Well, thank you, Mahmoud. Oh, yeah. Okay. Thank you, Mahmoud. I really appreciate it. Great talk, great demo, and I'm glad to see segmentor being used because, as you and I were chatting earlier, this is like first. It's been iterating a while. It just takes a while to kind of work out all the things.
Yeah, indeed.
But yeah,
Yeah, thank you very
very
much.
very interesting.
Yeah.
All right. Thank you. Bye.